أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم رب أدخلني مدخل صدق وأخرجني مخرج صدق واجعل لي من لدنك سلطانا نصيرا آمين يا رب العالمين اللهم صل على محمد وآله الطاهرين. This video is English language version of my previously distributed Persian language lecture on this topic, which I uploaded on Apparat website. Today I'm going to talk about enzymes. But what are enzymes? Enzymes are called biological catalysts. What is a catalyst? A catalyst is a substance which accelerates a chemical reaction. Actually, it increases the rate of a chemical reaction. It is not modified in a reaction. Actually, it is recycled in a chemical reaction. Enzymes are called biological catalysts because they catalyze biochemical reactions. They are involved in breaking down of previously formed biomacromolecules and in biosynthesis of new biomacromolecules. So they are involved in catabolism and anabolism respectively, and therefore in metabolism. But what is the quality of an enzyme? According to the roles of the proteins, they are classified into two major classes: structural proteins and functional proteins. Structural proteins are those proteins which build the structures of cells, tissues, and organs. But functional proteins are those proteins which have a function. Enzymes are functional proteins because they catalyze biochemical reactions. Actually, most of enzymes are proteins, but there are some exceptions for this rule. For example, there are some RNAs which catalyze biochemical reactions, so some RNAs are enzymes. Enzymes act on a molecule or on a substance which is called substrate. Sometimes an enzyme combines two substrates to form a larger molecule. Sometimes an enzyme breaks down one substrate to form multiple products. Enzyme acts on substrate to form enzyme-substrate complex. Enzyme-substrate complex is undergone some change and products are formed. The enzyme molecule is released, and again it is used in the biochemical reaction. K1, K2, K3, and K4 are rate constants. If K1 is greater than K2, the reaction proceeds toward the association of enzyme and substrate molecules and production of enzyme-substrate complex. If K2 is greater than K1, the reverse reaction proceeds toward the dissociation of enzyme-substrate complex. In the second reaction, if K3 is greater than K4, again the forward reaction takes place toward the formation of product. If K4 is greater than K3, the reverse reaction proceeds. But commonly, the K4 value is very negligible. But how an enzyme interacts with a substrate. Two models have been proposed for enzyme-substrate interaction. The first model is lock and key model, which was proposed by Emil Fischer in 1894. And the second one is induced fit model of Danilel Koshland, which was introduced in 1958. In the first model, the structure of substrate is complementary to the structure of active site of respective enzyme. In this model, substrate molecule acts like a key which is fitted to the active site of respective enzyme, which acts like a lock. Actually, substrate molecule is fitted to the active site of an enzyme like a key is fitted to its respective lock. This model also explains well the specificity of an enzyme for its substrate, but cannot explain the kinetic behavior of enzymes. So, in 1958, Danilel Koshland introduced his induced fit model. In this model, a structure of substrate is not complementary to the structure of active site of respective enzyme. But after binding of substrate to its respective enzyme, their structures become complementary. When substrate molecule approaches the active site of respective enzyme due to formation of intermolecular bonds, intermolecular interactions like hydrogen bonds, like van der Waals bonds, the structure of substrate will become complementary to the structure of respective enzyme. Please pay attention that size of substrate molecule is very smaller than that of respective enzyme. For better understanding, I drew the substrate molecule like a large key. But how enzymes accelerate biochemical reactions? I drew two reaction coordinate diagrams in the left for exergonic or exothermic reactions, and in the right for endergonic or endothermic reactions. X-axis or horizontal axis represents time course of reaction, and the y-axis or vertical axis represents free energy. Reactants should overcome an energy barrier which is called activation energy. Activation energy is related to a transient state which is called transition state. Transition state has least stability and highest free energy level. Transition state is a transient state in which reactant bonds are partially broken down and product bonds are partially formed. Blue curve represents uncatalyzed reaction in the absence of enzyme, and black curve represents catalyzed reaction in presence of an enzyme. As you see, enzymes reduce the activation energy of the chemical reaction. Actually, they stabilize the transition state of a reaction. in exergonic or exothermic reactions, the free energy level of products is lower than that of reactants. So exergonic reactions are spontaneous. They don't need energy and they release energy. Difference between the free energy level of products and reactants is called Gibbs free energy, which is shown with delta G. Delta G or Gibbs free energy is negative for exothermic or exergonic reactions because the free energy level of products is lower than that of reactants. For endergonic or endothermic reactions, as you see, the enzymes decrease the activation energy of biochemical reaction. Gibbs free energy for endergonic or endothermic reactions is positive because the free energy level of products is higher than that of reactants. So endergonic reactions are not spontaneous and they need energy. And at the end of this lecture, I would like to plan a question. Please use internet textbooks and other resources to answer the question. What is the meaning of zero value for delta G? والسلام على من اتبع الهدى.